Students, welcome you all on the behalf of Rahul Anand Tutorials. So as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually what? Whose name is actually the Rahul Anand Tutorials. So now let's begin with the very interesting topics which are often being asked in terms of your NEET, often the aims, often the All India Pre-Medical Examination. So these are literally belonging from which of the chapters as well as the topics of your book. So these are belonging from the sickle cell anemia. Now, so when we are talking about these cases, so you have to understand the certain facts in it. So let's understand about this in terms of the detail. So when we are talking about the sickle cell anemia, so these are what? So sickle cell anemia are the autosomal linked recessive disease as well as what? As well as the autosomal linked recessive disease. Now when we are talking about these cases, means how does this sickle cell anemia are being caused? So these are being caused by what? So these are being caused by the point mutation of what? So these are being caused by the point mutation of the beta globulin chain of the hemoglobin pigment, which are literally being found with reference to what? With reference to your blood. Now, so when we are talking about these cases, means what are actually the hemoglobin? Remember? So what are these? So hemoglobin playing an important role in terms of what? In terms to provide the coagulation properties with reference to what? With reference to your blood region. Often when we are talking about these cases, means what are actually the blood? Remember? So blood are the liquid fluid tissue. Often when we are talking about these cases, means what are actually the tissue? Remember what are these? So tissue are the group of cells. Now, as well as when we are talking about the different components of the blood, remember? So what are actually the different components of the blood? So we are having the plasma as well as the other fluid substances or either the other fluid components with reference to what? With reference to the substances generally present as well as being found with reference to what? With reference to your blood. Now, so when we are talking about the composition with reference to the plasma, so these are occupying with reference to the blood composition in terms of how much of the percentage? Tell me. So these are around 55%. Now, as well as when we are talking about the case with reference to the other viscous or other the fluid-like substances, so these are being employed with reference to what of the components? So it will be employed with reference to the here, meaning of the erythrocyte. Directly proportional for what? So directly proportional for the red blood cell. As well as you are very clear about that, means what are actually the red blood cell. Often its lifespan with reference to the human body is around how much? So its lifespan with reference to the human body is around 120 days. Now, after that you are often having what? After that you are often having the here in terms of the leukocytes as well as what? As well as the erythrocytes. As well as the leukocytes, as well as what? As well as the thrombocytes. Remember? So when we are talking about the case of the thrombocytes, so these are directly proportional for what? So your thrombocytes are directly proportional for the here main term of the platelets case. Now, after that you are often having what? So you are having the leukocyte directly proportional for what? So it will be the directly proportional for the white blood cell. Knowing as well as understood. Now, so when we are talking about the lifespan with reference to the white blood cell in terms of the body composition with reference to the human body, so it is around how much? So it is around here, meaning around 4 to 5 days. Literally very smaller lifespan when we are comparing the case with reference to what? With reference to the red blood cell. Often you are having the different components with reference to what? With reference to here, meaning in terms of the blood region. So these are what? So we have did about the red blood cell, then the white blood cell. Often you are having the here meant of the leukocyte, then what? Then the buffy coat. After that, when we are talking about the case of these minutum of what? Minutum of the white blood cell. So your white blood cell is often being further divided in terms of the broad categories as well as the red blood cell are oftenly being divided in terms of the broad categories. Now, so when we are talking about the case of what? Here in terms of the plasma. So it will be divided with reference to what? With reference to the case of the protein. Fine. So around 7% of the protein literally present with reference to what? With reference to the composition of the blood. After that, your protein are often being divided in terms of the 4 broad categories. So these are what? So these are the albumin, then the globulin, then the fibrinogen. So fibrinogen composition will be around how much? With reference to the protein region. Directly proportional coming under from the blood portion. So it will be around 7%. After that, the other fluid. Now, after that, in terms when we are talking about the case of the white blood cell, so these are often being divided in terms of the broad categories. So now, let's understand about this in terms of the detail. So when we are talking about the case in terms of the here main of the white blood cell, directly proportional for the erythrocytes. So these are often being divided in terms of the 3 broad categories. So these are what? So these are the lymphocytes, then the monocytes, as well as the neutrophils. So now, when we are talking about the case of the here main of the lymphocytes as well as the monocytes, so you are very clear about that. Remember, as well as understood what are actually the lymphocytes. So lymphocytes are playing an important role in order to provide as well as boost the immune system. Now, often you are very clear about that, means what are actually the immune system. So your lymphocytes are often being divided in terms of the 2 broad categories. So these are what? So these are the bilipocytes. Then what? Then the telepocytes. When we are talking about the bilipocytes case, so these will be doing what? So these are particularly being formed with reference to the bone marrow. So this will be formed where? So it will be formed within the bone marrow. As well as when we are talking about the case of the telepocytes, so it will be oftenly being formed with reference to the minute of the bone marrow. But when we are talking about the maturation case with reference to the bilipocytes as well as what? As well as the telepocytes. So the case is quite a bit different. So now let's understand about this. So when we are talking about the bilipocytes formation, so it will be particularly formed within the bone marrow, as well as the maturation will be often taking place with reference to the bone marrow. But when we are talking in the case of the telepocyte, the formation will be taking place in the bone marrow, but the maturation will be taking place in terms of the thymus. So you are very clear about that. Means what are actually the thymus? They are the lipoid system as well as the organs which are generally being included with reference to these. So we are having what? So we are often having the spelling as well as what? As well as these in terms of the when we are talking about the case of the lymphocyte system. Often when we are talking about these cases, means what are actually the thymus. So these are what? So these are oftenly the part with reference to these only in terms of what? In terms of the lymphoid organs. Often when we are talking about these cases in terms of the bilipocytes as well as the telepocytes, so your telepocytes are oftenly being divided with reference to the different composition of the cells. Remember? So these will be playing an important role to do what? So this will be playing an important role to provide the immune responses, such as what? Such as we are having the helper T cell, then the killer T cell, then the suppressor T cell, often the memorizing T cell. As well as when we are talking about these cases, so suppressor directly proportional for what? So it will particularly play an important role in order to provide the suppression mechanism. As well as when we are talking about the case of the memory T cell, so it will particularly remember the sequence of the event. So suppose that how the particular of the pathogen impact what? Impact our body, as well as the of the foreign agents literally come with reference to our body. As well as when we are talking about these cases in terms of the memory T cell, so you understood. Then the helper T cell will be doing what? So it will play an important role to maintain the coordination between these of the suppressor T cell as well as the killer T cell of the memorizing T cell. As well as when we are talking about these cases, means what are actually the autosomes. So you are very clear about that. Remember, as well as knowing. So autosomes are what? So any kind of the chromosomes which are directly proportional as well as not coming under what? So the chromosomes which are literally not coming under the sex chromosomes. So it will be coming under what? So it will be coming under the autosomes. So what are actually the sex chromosomes? So these are the X chromosomes as well as the Y chromosomes. Remember, as well as understood. So here many terms the X chromosomes directly proportional for responsible for what? So responsible for coordinating with reference to the femaleness characteristics. But literally when we are talking about the case of the Y chromosomes directly proportional for what? So these are directly proportional for the case of the dominancy. Often these are known as the dominant trait, coming under the dominant trait, as well as directly proportional for discovery as well as forming the maleness characteristics. Knowing now. So when we are talking about these cases in terms of the allsomes, so allsomes will be what? So it will be the sex chromosomes. But here we are having what? So but here we are having the autosomes. Now, often when we are talking about these cases in terms of the genes, so we'll be understanding about that later. So when we are talking about the case in terms of the sickle cell anemia, so you are having the two types of the facts as well as the terminates. So we are having the homozygous alleles as well as the heterozygous alleles. So when we are talking about the case of the heterozygous alleles, so we'll be having what? So we'll be having the HBA, then the HBS. So these are particularly could be the carrier of what? It could be the carrier with reference to the sickle cell anemia. But when we are talking about these cases in terms of the heterozygous alleles, so these are oftenly being playing an important role in order for what? In order for controlling the in terms of the defect, controlling from the defect of the sickle cell anemia. Fine. But when we are talking about these cases in terms of what? In terms of the in terms of the homozygous alleles. So homozygous alleles will be what? When two types of the alleles will be present, but literally it will be having the same types of the characteristics in it. Such as what? Such as the we will be having the HBS, and another will be what? So it will be often the HBS. Thus, in this case, we could say that what? So in this case, we could say that the homozygous alleles are what? So these are the carrier of the sickle cell anemia. So you are very clear about that. Means what are actually the alleles as well as the allelomorphs. So alleles as well as the allelomorphs are what? So the alleles as well as the allelomorphs are the different form of gene which are being localized where? Which are being localized with reference to the same locus of chromosomes. So you are often very clear about that. Means what are actually the chromosomes. Now, so let's understand about these. Means what are actually the role with reference to these in terms of the sickle cell anemia with reference to the protein. So you are very clear about that. Means what are actually the protein. So So when we are talking about the case of the protein, so proteins are what? Proteins are the macromolecules of the amino acid. But when we are talking about the case of here, when we talk the amino acid, so just reverse the process. So amino acid will be what? So this will be the macromolecules of the protein. Now, so you are very clear about that, means what are actually the protein, often being divided in terms of the four broad categories, as well as all the proteins are having what? So these are having the amino acid. So these are generally being formed with reference to how much of the amino acid. So these are being formed with reference to the 20 basic amino acid. Remember? So these are what? So these are the proline, then the valine, then the glutamic acid. Often what? Often you will be having the threonine as well as the histamine. Often what? Often the another of the components, such as what? Such as these in terms of the proline. So we'll be understanding about that later. Now, so when we are talking about these cases, when we talk the proteins, so all of the basic proteins are generally being composed with reference to what? So all of the proteins are generally being composed with reference to the 20 of the functional. Here, when we talk the amino acid. Fine. As like we are having what? As like we are having the 26 letter with reference to our English alphabet. Now, so when we are talking about these cases, so here we will be having the particular sequence. So just understood about this sequence. So we will be getting what are actually the defects are generally being caused with reference in the case of what? In the case of the sickle cell. So when we are talking about the composition with reference in the case of the sickle cell anemia, so we'll be having what? So we are having the vellus, leukotherium, then what? Then the pro-glu-glu. So in this case, what will be happening? All the overall sequence will be same. But literally, when we are talking about the case in terms of what? In terms of the sixth position. So sixth position will be employed by what? So your sixth position will be employed in terms of what? In terms of the proline. So primarily there was what? In terms of the normal of the what? In terms of the normal RBC. So we'll be having the sequence of the protein in terms of the glutamic. Fine. In terms of the glue. So here glue is what? So here glue is the triplet codon as well as the multiple codon. Remember about the case of the codon. So here codon was particularly being demonstrated as well as discovered by who? So it was discovered by the George Gangue. So George Gangue particularly played an important role for what? So he was the scientist, played an important role to discover as well as propose what? As well as propose the multiple codon system as well as the triplet codon system. So there was particularly the series of experiment as well as what? The discoveries as well as what? As well as the experiment as well as the research were performed. And finally, in terms of this, in terms of the triplet codon, these proteins in terms of what? Were named. Fine. As well as thus we are getting here many terms the names such as the pro, then what? Then the glue as well as the here many terms the threw, then what? Then the hista, his fine. So here in this case we will be having what? So the sixth position of the glue will be represented as being replaced by what? So it will be replaced by what? So these are particularly replaced with reference to the valine case. So you could often find it on the board here. So when we are talking about the case of the sickle cell anemia, so these are what? So these are the here many of the genetic disease of what? Of the autosomes represented by what? So these are literally represented with reference to the here many of the HBS. Often when we are talking about the case of these many of the sickle cell anemia. So when we are talking about the case of the sickle cell anemia, so these are what? So sickle cell anemia are the genetic disease of what? So sickle cell anemia are the genetic disease of the autosomes, which are particularly being represented with reference to what? With reference to the HBS. Often when we are talking about the case of the sickle cell anemia, so these are being caused by what? So these are being caused with reference to the here many of the gene mutation. So the replication of gene is very accurate, and you are very clear about that, means what are actually the accurate. But due to some of the external as well as what? As well as the internal hindrance, what will be happening? There will be change with reference to the regular frequency of the gene which will be employed as well as viewed. So by this mechanism, what will be happening? So by this mechanism, the gene mutation will literally be produced. As well as you are very clear about that, means what are actually the mutation. Remember? So you are having the name of the scientist such as what? Such as the Hugo de Verris. So your Hugo de Verris played an important role in order for what? In order to propose the mutation theory. Often you are very clear about that, means what are actually the sequence of events are generally taking place in terms of the case of the. So when we are talking about the case of the sickle cell anemia, so these are being caused with reference to what? So these are being caused with reference to the gene mutation. Often these are being controlled by what? So these are being controlled by the here many of the beta chain of the HB. So you are very clear about that, means what are actually the beta chain of the HB. HB here directly proportional for the another cases. So we'll be understanding about that later. Often, so you are very clear about that, means what are actually the alleles. Fine. So alleles and allelomorphs are what? So these are the different form of gene which will be localized where? Which will be localized on the same as well as the different locus of chromosomes. So primarily, which will be localized with reference to the different locus of chromosomes coming under the alleles as well as the allelomorphs cases. Often you are very clear about that, means what are actually the gene mutation. Remember what are actually the gene mutation. So the replication of gene is very accurate, and you are very clear about that, means what are actually the accurate. But due to some of the external as well as the internal hindrance, what will be happening? There will be change with reference to the regular frequency of the gene which will be employed as well as viewed. So by this mechanism, what will be happening? So by this mechanism, the gene mutation will literally be produced. As well as you are very clear about that, means what are actually the mutation. Remember? So you are having the name of the scientist such as what? Such as the Hugo D. Verris. So your Hugo D. Verris played an important role in order for what? In order to propose the mutation theory. Often you are very clear about that, means what are actually the sequence of events are generally taking place in terms of the case of the mutation. Remember? So prior to the gene, thus when we are talking about the case of the mutation, remember, as well as understood. So primarily there will be what? So primarily there will be the change in terms of the single gene. After that, what will be happening? After that, there will be the change with reference to the here, in terms of the DNA. After that, the change in terms of what? After that, there will be the change in terms of the mRNA codon. After that, what will be happening? After that, there will be the change in terms of the here, in terms of the sequence of the amino acid. And after that, there will be the change in terms of the protein. And after that, what will be happening? And after that, there will be the change in terms of the characteristics. Then there will be particularly change are literally being viewed with reference to what? With reference to the phenotypes. And after that, what will be happening? So when there will be particular change in terms of the phenotypes are literally being organized as well as being found. After that, the mutation case are literally being present as well as employed. So you are very clear about that, means what are actually the phenotype. Remember? So we are often having the two type of the ratios. So these are what? So these are the phenotypic ratio. Then we are having the genotypic ratio. Calling as well as understanding about the means what are actually the phenotype directly proportional for the physical appearances, as well as when we are talking about the case in terms of the genotype. So it will be what? So it will be directly proportional for the appearances with reference to the gene expression, or either what? Or either expression with reference to the chemical level. And you are very clear about that, means what are actually the chemical level generally being present with reference to our body. Now, so you are very clear about these, means what are actually the different types as well as the parts of what? Of the here, made of the mutation. So these are what? So these are the chromosomal mutation. Then we are often having the gene mutation coming under these case of the sickle cell anemia. So you will be understanding as well as causing, as well as understanding what are actually the cause of the sickle cell anemia. So what are actually the cause? So the cause are generally what? So these are the gene mutation. Fine. As well as when we are talking about the case in terms of the structure of the normal cell with reference to what? So when we are talking about the case of the normal cell in terms of the RBC cell. Fine. So this will be what? So these are particularly resembling the here in terms of the biconcave shape like structure. But literally when we are talking about the case of the sickle cell anemia, so it will be resembling what kind of the structure? So it will be particularly resembling the here in terms of the concave shaped structure. Often when we are talking about the concave shaped structure, so these are what? So the I-shaped structure are generally coming under the here in terms of the concave shaped structure. Thus, due to these features, in terms of what? In terms of the concave nature, what happens? The fine amount of the capillaries in terms the oxygen could not be passed out. Thus, these are often coming under what case? Thus, these are often coming under the case of the bloodlessness symptoms. So anemia are directly proportional for what? So your anemia are directly proportional for the bloodlessness condition. So you could often find popularly on the board. So we are having the two types of the structure with reference in terms of the normal RBC. Then what? Then the defected RBC. So the defected RBC are countered by what? So these are being arrested as well as countered with reference to the sickle cell anemia. Find here on the board here. So this is the normal cell of the red blood cell in terms of the arthriocyte. Then you are having the concave shaped structure with reference to what? With reference to the defective cell in terms of the red blood cell. In terms of, so this will be encountered with reference to the sickle cell anemia. So this is about the condition when we are talking about the case of the sickle cell anemia, in which we have dealt about the certain of the features. So you could often write these. So if you like the work with reference to the education as well as the lectures, as well as what? As well as the videos. So you could often what? So you could often like, subscribe as well as share my lectures. Often subscribe below in the here many of the lecture content as well as what? As well as share my lectures with reference to what? With reference to the Rahul Anand tutorials. So please like, subscribe as well as share my lectures. So very thank you.